Let's take a minute and talk about the structure of viruses. Viruses can be classified as either being naked or enveloped. Naked viruses consist of the things we've already talked about. A piece of genetic material, either DNA or RNA, surrounded by a capsid made out of protein. Many of the plant viruses, most of the bacterial viruses, are this naked type. Enveloped viruses are different. Enveloped viruses are a piece of genetic material surrounded by a capsid, and then surrounded by a layer of membrane, which is referred to as an envelope. The envelope is actually not of viral origin. The envelope is actually acquired from the host cell, from the plasma membrane of the host cell, when that viral particle exits. Now we'll talk about how a viral particle exits a cell a little bit later on. Most of the animal viruses are the enveloped type. So it's a nucleocapsid surrounded by a piece of membrane that was essentially stolen from the host cell. Many viral particles are also covered in surface protein spikes. Y'all have seen this very famous picture of the SARS-CoV-2 virus. This is the picture that all the news organizations use all the time when they talk about this virus. And these red structures that are sticking off of the surface, these are protein spikes. So the SARS-CoV-2 virus is an enveloped virus, and it also has these spike proteins coming off the surface. Now, while the envelope comes from the host cell, the spike proteins are actually viral. So they come from the code in the viral genetic material. It's the spike proteins that bind to receptors on the host cell membrane, and these are especially common in animal viruses. You do see some spikes on other types of viruses, plant viruses, etc., but they are especially common in animal viruses, as are envelopes. On this slide, you can see some drawings of the two kinds of viruses, the naked viruses on the left and the enveloped viruses on the right. So what the artist is showing us here on the left, up at the top, is a cutaway view of a viral particle. We're peeking inside of a nucleocapsid. You can see the genetic material. It's depicted in purple. And then you can see this sort of coating, this shell of protein depicted by these little yellow balls. This coating structure is referred to as the capsid. They're also showing us that this naked virus has spikes on it, shown in purple. Over on the right-hand side, you can see an enveloped virus drawn. Again, the artist has cut away a piece of the virus so that we can peek inside. You can see again the purple genome, the genetic material. You can see the yellow capsid, the protein capsid. And then you can see this sort of purpley gray envelope around that structure. Remember, this is just a piece of cell membrane that became part of the viral particle when it exited the host cell. And then you see these blue structures sticking through the envelope. Those are spike proteins. Down at the bottom, they've also drawn a naked virus on the left and an enveloped virus on the right, but just to show us that not all viruses have that sort of round spherical shape. This virus down at the bottom is quite elongated. but it still has the same basic structure. It has genetic material in the middle, surrounded by a protein capsid. Almost looks like corn on the cob in this drawing. And over here, an enveloped form of this virus, which has, again, genetic material in purple, a protein capsid, or at least part of a protein capsid in yellow, and then a membrane envelope surrounding that. Capsids are composed of protein, and for many viruses, it's a single kind of protein that repeats over and over and over and over again to build this capsid structure. The individual subunit protein that you find in a capsid is called a capsomere. So again, many viruses have only one kind of protein in their capsid. So they only have one type of capsomere, but they have many copies of that capsomere surrounding the genetic material. Evolutionarily speaking, having only one or maybe just a couple of different capsomere proteins would be a useful trait to have. Because remember where proteins come from. Remember the central dogma. DNA contains genes, and genes give rise to proteins. So if you need fewer proteins to build your capsid, you will have fewer genes in your genetic material, and that just is a simpler structure to have, easier to replicate, better for you overall as a viral particle. Take a look at the picture down here on the right-hand side. This is a drawing of a type of virus called the tobacco mosaic virus. This is a virus that, you might guess, infects the tobacco plant. And what the artist has done is drawn the genetic material in red and the capsomere protein as this blue sort of a rod-shaped structure. So the artist has taken away some of the capsid so that we can see the genetic material. And in this type of virus, this elongated shape of virus, the genetic material just sits in the center like a long helical structure. And the protein capsid surrounds that helical structure. The tobacco mosaic virus, like many viruses, has only one kind of protein in its capsid. It has 2,130 copies of that one kind of protein. So each one of these turquoise blue structures that you see drawn in this capsid, that's all the same kind of protein, just many, many copies of it, over 2,000 copies of it to build this capsid. The tobacco mosaic virus happens to use RNA as its genetic material, and it happens to use one strand of RNA. So this red helical structure is a single strand of RNA that is found in the center of that virus. So there are helical or elongated shaped viruses, but there are other shaped viruses as well. The helical kind of virus gets its name not so much because its genetic material is in a helix, but because the capsomere proteins that create the capsid, they wrap around that virus in a helical manner. Let's go back to the tobacco mosaic picture for a minute. If you were to follow these turquoise molecules, if you were to follow them, they actually wrap around this viral particle in a helix, in a helical way. Very, very organized, very symmetrical, a common feature of viral particles. So there are helix or helical shaped viruses. There are also polyhedral viruses, And these are viruses that from a distance look like spheres, but when you get up close to them under the microscope, they're actually a polyhedron. They are composed of triangular shaped plates that are joined together in a spherical shape. Then there are the true spherical viruses. And finally, there are what we call complex shaped viruses. There are really only two of these that you should be familiar with. There's the shape of the kind of virus that infects bacteria. Those are called bacteriophages. And there are what are called giant viruses. And we'll take a look at all of these shapes. On this slide, you can see the range of viral shapes. Up at the upper left-hand corner, you can see what is a polyhedral shape. This happens to be a depiction of an adenovirus. This is the entire virus. This is the whole capsid with the genetic material tucked inside. So we can't see the genetic material in this picture. Notice, though, that there are these triangular shaped flat plates on this structure. Several of them that all come together and join to make this polyhedral structure. There are also proteins, spike proteins, coming off of this virus. This is a depiction of an adenovirus. This is a depiction of the influenza virus, and this is a true spherical virus. It also has spike proteins coming off of the surface, but that true spherical shape comes from the fact that there's an envelope around this virus. Here's that helical shape again. Again, this happens to represent the tobacco mosaic virus. So these are the more sort of basic shapes that viruses come in. This is one of the complex shapes. So this is a depiction of a bacteriophage. And that word bacteriophage just means a bacterial virus. There are lots of different kinds of bacteriophages. It's just that they all have the same shape. Now look at this thing. It doesn't look like it ought to exist in nature. It has a very complex anatomy. But this is actually what a bacterial virus looks like under the microscope. So when you look at a bacteriophage, you can describe different pieces of it, different parts of its anatomy. It has a head structure, as you can see in this actual electron microscope image. It has a head structure, and that's where the genetic material is found. It has a neck structure. It has what's called a sheath or a tail sheath. And then it has these spiky, what looks like legs that are referred to as tail fibers. This is just a drawing, again, of a bacteriophage. And you can see that the head actually has a polyhedron shape. And again, then you get this very strange sort of a neck structure before you hit these spidery tail fibers. Bacteriophages actually use chaperone proteins so that when they're being assembled inside of the host cell, they are assembled correctly into this complex shape. Recall that a chaperone protein is a protein that chaperones or watches over the folding of a protein. So inside the virus's genetic material, there is code to build a chaperone protein among all the other types of proteins. And this chaperone protein, when it gets produced by the host cell, will guide the folding of this very complex shaped bacteriophage. Now, the other kind of complex shaped virus that we know of is called a giant virus. Now, this virus actually has a fascinating story because it was only discovered very recently. It was discovered in the early part of the 2000s. For many years, scientists had seen this virus underneath the electron microscope, but they weren't quite sure what it was. And because of its size, they assumed that it was some kind of either a bacterium or maybe an archaeal organism. If you look at this virus under the microscope, it doesn't look like any of the other viruses. It's too complex. It's also quite big, relatively speaking. So for many years, people wondered exactly what it was, didn't really know what it was. And it wasn't until, again, the early 2000s that these viruses were isolated and grown in the laboratory and further characterized. They are viruses, they are not cells. And the only ones we've discovered so far are only capable of infecting amoeba. Amoeba, remember, are one of the single-celled eukaryotic microbes. So this giant virus is the type of virus that infects amoeba. There are several of them discovered so far, and they go by names like mimivirus, mamavirus, and megavirus. The mimi and the mama and the mega stand for something. In other words, they're abbreviations. We don't know if there are giant viruses that are capable of infecting other kinds of cells. We just don't know. But so far, the ones we've discovered infect amoeba. Very interesting group of viruses. On this slide, you can see a range of viral shapes, both as drawings up at the top and then as images taken with an electron microscope. So up at the top here, this is a picture of our friend, the tobacco mosaic virus. This would be the entirety of the virus, so you could call this a nucleocapsid. There's genetic material inside. Remember, that's RNA for this virus. And each of the blue circles that you see on the outside, that's a capsomere. And all of the capsomeres together make up the capsid, the protein coating. This is what this virus looks like underneath the electron microscope. Now I want you to notice its size. We're talking a very, very small particle in the tens of nanometer range. Next door to it, there's a polyhedral shaped virus. The artist has cut away one of the triangular plates so we can see the DNA inside. This again represents an adenovirus. The little purple circles that you can see, each one of those purple circles is a capsomere, and the whole thing is the capsid. The whole purple shell is the capsid. The entire particle would be called a nucleocapsid. This is what adenoviruses look like under the microscope. They almost don't look real because they are so just perfectly symmetrical, especially with those spikes coming out of them at regular intervals. Again, very, very small particle. Next door to that, we have a drawing of the influenza virus. They're showing us this purple structure in the middle here. This is the capsid. Remember, influenza virus is an enveloped virus. So the orangey structure on the outside, that's membrane. That's the envelope. So the capsid is actually inside the envelope in purple, and that surrounds the genetic material. And they're small, but you can just make out the purple spike proteins on the surface of that virus. This is what influenza looks like under the microscope. It almost looks like our friend, the coronavirus, in that the coronavirus is also enveloped and it does have spike proteins. But notice that there are lots of spike proteins on the influenza virus. Many, many, many more than you see on the coronavirus. And lastly, we have a bacteriophage, an artist's drawing and an actual image. Look at this thing. It looks like an alien creature. And it is absolutely just fantastic in its shape. So when you think about viral structure, one of the key things to remember is how big they are. Generally speaking, if you look at all viral particles, they range in size from about 30 nanometers to about 300 nanometers. Okay, very, very small. The picture over here in the upper right is drawn to remind us of just how small viruses are. This is a depiction of a cell, and it's a eukaryotic cell. So this large orange circle represents a generic eukaryotic cell. Here in purple, this is the nucleus. And you can see other organelles floating in the cytoplasm. Next to it is a representative drawing of a bacterial cell, a rod-shaped bacterial cell, much smaller than the eukaryotic cell. Over here in the blue, these represent viral particles. So much smaller than a bacterial cell. And remember how hard it is to see bacteria under the microscope. They are still pretty small, even under a 100x objective lens. Bacteria are still pretty small. The viruses are much smaller. The giant viruses are the exception. That's how they got their name. Giant viruses range in size from 400 to about 1500 nanometers. And 1500 nanometers is the same as 1.5 micrometers. So the largest of the giant viruses are the same size as the smallest of the bacteria. You can understand why scientists didn't know they were looking at a virus for a long time. So, some of the cocci that we've looked at under the microscope are about 1 micrometer across. So that would be, approximately the same size as the largest giant virus. And remember, 1 micrometer is 1,000 nanometers. So most viruses are much smaller than a bacterial cell. Remember, remember, remember. Viral particles cannot be seen with a light microscope. It's not that you can't see them very well. It's not that you can't see them in much detail. You cannot see them under a light microscope, with the exception of the giant viruses. I put this picture in because this is an artist's depiction of just how small viruses are. The artist has drawn a section of the membrane and the cell wall of a gram-negative bacterial cell. So just a little piece of the membrane and the cell wall. This is the cell membrane down at the bottom. So this would be the cytoplasm here. This is the outside of the cell right here. You can see that the membrane has blue heads on either side and yellow gold tails in the middle. You can see purple proteins embedded in the membrane. You can also see this gigantic structure. This represents a flagellum. That's how large a flagellum is compared to the phospholipids in a membrane. This grayish layer here represents the thin layer of peptidoglycan that's in the cell wall of gram-negative bacteria. And then you can see the outer membrane that we find on gram-negative bacteria. And these structures that are coming off of the outer membrane, this represents LPS, lipopolysaccharide. Remember, the bacterial cell benefits from LPS because it strengthens the cell wall, the outer membrane. So the reason the LPS is there is because it's useful to the bacterial cell. But when LPS gets set loose in the human body, it's toxic. It's toxic to us. So remember, bacteria don't make LPS as a weapon against humans. It's just part of their structure. It's a helpful part of their structure. It just so happens that when it gets released, it's toxic to us. There's also a representation in the drawing of a pilus here. This is what I want you to notice. Right here. That's a bacteriophage. Here's another one. Here's a polyhedral virus drawn. Here's another polyhedral virus drawn. The artist is trying to show us that even on this little tiny piece of membrane, these viruses look very small. Much, much smaller than the entirety of the bacterial cell.